This, therefore, this wide variety is available. The composition is not exactly the same. It is constantly changing, and there is a fluidity in that. It is a dynamic one, and it is also fluctuating during different stages of cell development. Uh, Particularly, the amount of cholesterol that can change uh, at variety of time, and this change in the composition that will affect or that will alter the fluidity throughout the development stages. The materials which are incorporated into the membrane or they are deleted or they are added like that. So there is a fusion of intracellular vesicles in the membrane. We have seen that as exocytosis or endocytosis. Both processes taking place. That is, or even if you remember last year, all the modes of engulfment, phagocytosis, pinocytosis, exocytosis, endocytosis. These all things, because of the fluidity, this membrane can do. If the membrane is a continuous with a tubular structure, then the tube can be drawn into the membrane continuously. The concentration of membrane components in aqueous phase is low uh, because the stable membrane components have low solubility in water, and there is exchange of molecule between the lipid and aqueous phase. You have studied this as a part of absorption. Now, what are the lipids which are present? You have seen again structure of these lipids earlier. These are important. Two lipids are comparatively important. Three lipids are there. Other phospholipids, glycolipid, and sterols. They are there. These lipids are antipathic, means having both hydrophilicity and lipophilicity. If you remember, again, absorption distribution, absorption particularly, and fluid mosaic model of cell, then you will remember that there are polar head, non-polar head, and because of this polar and non-polar head, this is happening. This is there. Then next is the fatty chains which are present in the phospholipids and glycolipids. They contain even number of carbon atoms between 16 to 20, and 16 to 18 carbon fatty acid are the most common. They are saturated or unsaturated. The fatty acids are, and whenever there is a double bond, they are most of the places cis. If you remember again organic chemistry, cis and trans. So they are cis. They have profound effect on membrane fluidity. As the unsaturated lipid, they create a ink, prevent the fatty acid from packaging together as tightly. They produce a slightly thickness a gap type situation, and they decrease the melting temperature. They increase the fluidity of the membrane, and there is the ability of some organism to regulate the fluidity of their slate membrane by altering lipid composition. And that particular situation by altering the fluidity of this alteration of fluidity because of the change in the composition, lipid composition, is also called as a homeoviscous adaptation, that is change in the fluidity. This is also one of the mechanism of homeostasis body is doing, but this is quite having higher depth, which you might have not studied earlier. The entire membrane is held together by non-covalent. There is no covalent bonding, therefore it can be separated easily with some higher portion. So it is non-covalent hydrophores are there. It is structure is quite fluid. It is not flexly tightly in a place. Under physiological condition, phospholipid molecule in the cell membrane, they are in a liquid crystalline state. That is, liquid molecules are free to diffuse and exhibit rapid lateral diffusion along with the layer. And the exchange of phospholipid molecules between intracellular and extracellular is possible. Rat, you might have seen the rats are there in various rivers, or you can check in images or videos. We say that it is a water rafting, that is in water, small single person, small boats are there. They are called as rafts. So similarly, this lipid rafts and caveolia, these are the example of cholesterol entries, microdomains in the cell membrane, and the fraction of lipids, they are in direct contact with the integral membrane protein. So we call them as lipoproteins also. Then they are tightly bound protein surfaces. They are called as a annular lipid cell, and it behaves as a part of protein complex. So this all this structure, they are playing important role. Cholesterol is also there, or sterols. We have already seen third part, lipoprotein, glycoprotein, and third one is uh, sterols. So cholesterol is dispersed in varying degrees throughout the cell membrane. Means it is in like a patches in the irregular spaces between the hydrophobic tails of the membrane lipids. It confers stiffening and strengthening effect to the membrane. Because of that, whatsoever exactly fluidity test is slightly reduced. The amount of cholesterol in the biological membrane varies between organisms, cell types, and individual cells. So, different cells will have different composition of cholesterol. It regulates the fluidity of overall membrane. Cholesterol controls the amount of movement of these cells. At high temperature, cholesterol inhibits the movement of phospholipid fatty acid chains, and thus it reduces the permeability. Then the cholesterol in cooler temperature, the opposite action is seen, that is, it increases the permeability. The cholesterol production can be upregulated in response to cold temperature, while vice versa. It acts as antifreeze. Cholesterol maintains fluidity of membrane, we have already seen, and it is more abundant in cold weather animals than warm weather animals. In plant also, which lacks cholesterol related compounds called as sterol to perform the similar function. It is not cholesterol but other sterols are there. Then phospholipids, they are forming lipid vesicles. Example of this is a liposome. You might have studied earlier as a, one of the organelle. They are used in laboratories. Yes, liposomes are one of the pharmaceutical situation which is used for the delivery of drug. Okay, so drug is incorporated in this phospholipid and thus it is delivered. So these liposomes are spherical pockets with this similar lipid bilayer. They are particularly the used for drug delivery and other things. They are there. In pharmaceutics, these liposomes are studied well. They are formed by suspending lipid in. Will not go into this because it is a liposome is totally a pharmaceutics matter. The next important components are the carbohydrates. These carbohydrates are bound with the proteins, so we call them as a glycoproteins. Some are bound with the lipids, so we call them as a glycolipid. Example of these are cerebrocytes, gangliocytes. Whenever sites are there along with that means glycosides. Okay, so there are many sites you may encounter in pharmacognosy, etc. There are many glycosides are there. Carbohydrates are important, particularly in cell-to-cell recognition. They help in as a receptor. They are located on the surface of cell where they recognize host cell and share information. Viruses bind to cells using these receptors. They causes infection. Most part of no glycosylations occur in the membrane of the cell. Generally, glycosylation occurs on the extracellular surface of the plasma membrane. There is a one structure that is a glycocalyx. It is an important feature of all cells, particularly epithelium, having microvilli. That is, microvilli are, you remember, you might have studied in intestines. The glycocalyx participate in cell adhesion, lymphocyte homing. Cell adhesion is very, very important part in immunity because these are the various adhesion proteins are there, and these adhesion molecules, they play important role for anchoring of the cell and all. Lymphocyte homing is also similar concept in immunology and many other. See, one thing I would suggest that, see, when we are going for this higher depth, every places you have to note down. These are the additional function which you might have not seen earlier. Why I am telling this? Because now we are understanding role of carbohydrate in immunology. How this carbohydrate which are present on the cell membrane, they participate in the immunology. What importance they have. Now, when we go ahead in future, a very important thing is that the simplest thing which we have to take home a message from this is uh, these all are the complex and specific structures, whether it is carbohydrate, whether they are protein, or whether the lipids are there. And because of that, because of their structures, they are participating in important functions. Okay. Now, any structural modification. Now, why there can be structural modification? Because of any genetic change, directly or indirectly. So, any structural modification, if it is happening, that will cause, or if any change in the amount of formation is happening, increase or decrease. So, anything which is, you can note it down, that is anything which is either increase or decrease in the amount, anything which can modify the structure, okay, alter the structure. So, any alteration from the normal population or usual population, if takes place, then that is creating a pathology. Okay, this pathology, if it may be treated with the drugs on direct impact whatsoever happening, or it may not be treated. Example, in some cases, example of this carbohydrates, because of that some receptors are overactive, or there is upregulation, then we try to put in some antagonist, okay, so that we can block the receptor and we get the normal type of activity. But maybe in future we'll not go for using this kind of antagonist. What we'll do is wherever it is there, we'll try to correct it genetically. Maybe in future the action will go in this way. And therefore, these functions understanding plays important role. Why we are trying to understand is what are these structures and why, how they are participating in various activities. So just now we have seen that these glycocalyx participate in cell adhesion, lymphocyte homing, etc. And because of that, that means that it has important role in cell adhesion or lymphocyte homing. The penultimate sugar means highest sugar is galactose, and the terminal sugar is the sialic acid. And these are, again, you might have studied all this in biochemistry. It is modified in the Golgi apparatus. Sialic acid carries a negative charge, and it providing an external barrier. Again, if you remember cell membrane last year, you might have studied. There are two important things which are happening. One is electrochemical gradient, and second is a concentration gradient. So there are changes in the concentration across extracellular field and intracellular field. So this is one barrier, that is from higher concentration to lower concentration. If pores are there, if passive diffusions are possible. And second thing is the electrochemical gradient, that outside it is positive, inside it is negative. So this carries a negative charge. It provides an external barrier to the charged particle. Now proteins, very important thing is there in proteins. We have got a variety of proteins. We classify them into three important classes, that is integral proteins, then lipid anchor proteins, and peripheral proteins. Okay. Now in that, if we attach sugar, then we call them as a glycoprotein. If we attach, or if the sugar is attached, then glycoprotein, lipid is attached, lipoproteins are there. Okay. So these three important proteins are there. What are their? Let us have a brief discussion about it. What do you mean by integral proteins? So these are integral proteins are, they are present on the membrane at distances. They have hydrophilic cytosolic domain, that is the domain towards cytosol. is hydrophilic in nature, that interacts with the internal molecules, and hydrophobic membrane spanning domain is there that anchors it within the cell membrane. So that is it is put into the, or it is embedded, we can call it as. So it is embedded into it. The hydrophilic extracellular domains are there. They interact with the external molecules. The hydrophobic domain consists of one, multiple, or combination of alpha helices and beta sheet protein motifs. Now this gives you all information about various receptor we have seen. Okay, so what are the example of these? Are all various types of ion channels, proton pumps, G protein coupled receptor. So whatsoever kind of transporters are there. Now those who are interested, last year it was there, but you can see in Goodman and Gilman whole chapter is provided on transporter proteins. Okay, it will help you particularly in future, that how important these transporters proteins are there. There are very good classification it is given, various SLC type of transporters, ABC transporters. Okay, so variety of transporters are there. So similarly, we know various types of receptors are there, various types of transporters are there, and many transporters. Just now, recently in diuretics, we have seen that very at every segment of the nephron, we have seen variety of transporters. Okay, at some places there is sodium potassium import is there. At some places sodium hydrogen exchangers are there, or contrast ports are there. At some places only water is taken in. So we have seen that there are variety of such channels or pores or things are present, and therefore they are important in diuretics or in physiology of urine formation. Just a small portion of kidney, or a very small unit, that is the smallest unit of kidney, nephron, in its very small structure at various places, variety of transporters are present. Okay, and they are a very specific area wise. We have seen that if this be modified with the drug, or if there is an inherent problem, that will affect the kidney function. So similarly, these channels are there. We have seen proton pump, where hydrogen is there. We have seen various receptors. G protein is one example, but there are many examples are there, and all these examples are part of integral protein. So may it be G protein coupled, may it be the enzyme linked receptors. All these are there. Then other are lipid anchor proteins. They are covalently bound to the single or multiple lipid molecules. They have hydrophobically inserted into the cell membrane, and they anchor the protein. The protein itself is not in contact with the membrane. Example of this is the G protein. Again, G proteins. I am not calling G protein coupled receptor. At the end, the G protein is there. When receptor is activated, then G protein, GI, GQ, GS, they are there. They are activated. So these are the lipid anchor proteins. They are anchored by lipid, and when this receptor is bound with the ligand, then this causes changes through mediated the action through G proteins. Then next one are the peripheral proteins. Peripheral proteins, they are attached to the integral membrane proteins. They are associated with the peripheral regions of the lipid bilayer, and these proteins tends to have only temporary interactions with the biological membrane. Once reacted, the molecule dissociates to carry out on its work in the cytoplasm. So example of these are the enzymes, hormones, some of the enzymes, some of the hormones. These are the peripheral proteins which are acting accordingly. So this is all the action which are there. Apart from that, we can have many other functions the cell membrane is carrying. These are some of the function. If you enlist what are the function these proteins and all these are carrying, their electrochemical gradients are there, and then concentration gradient is there. Because of all this gradient, accordingly it will be acting. Then next we have seen the electrochemical behavior it is causing. Example of this is the neurotransmission, or the impulse conduction, or skeletal muscle contraction. So this is again another example of that. Then this formation of these proton pumps are there. Then they are taking place or they are forming these acids. Then G protein already we have seen. So these are various activities are there. Apart from that, this cell membrane which are exposed to the outside environment because outside portion faces the extracellular fluid. So they are important site for cell to cell communication. Large variety of protein receptors and identification proteins we have seen, and they are there. And antigens are also present on the surface of the membranes. So function of the membrane proteins can also include cell-to-cell contact, surface recognition, cytoskeleton contact, signaling, enzymatic activity, and transportation of the substances across the membrane. These are the variety of activity this membrane is doing because of the protein which are present. Most membrane protein must be inserted in some way into the membrane, and this occur the N-terminus signals, that is signal sequence of the amino acid directs proteins to endoplasmic reticulum, where these proteins are inserted into lipid bilayer, and these proteins are then transported to their final destination in vesicle, and then vesicle will fuses with the target membrane or places. We have already seen vesicle formation earlier. So what are the functions? There are many functions are there, and because of that, cell membrane becomes very important into this. These are cell membranes. They are surrounding the cytoplasm, so it is separating intracellular and extracellular environment. Now, because of that separation, whatsoever pharmacological actions are there that we have seen as a electrochemical thing or repolarization, etc. We have seen passages of ion because of that. What happens? We have seen muscle contraction. Many things we have seen because of this particular situations. Okay. Now, thing is, uh, the cytoskeleton is there additional benefit is so anchoring the cytoskeleton to provide the shape of the cell. It attaches the extracellular matrix and other cells to hold them together, and they form the tissues. Again, for tissues, you can understand, you can go through the your Tortora or any physiological book. You will find various types of junction, tight junction, gap junction, etc. So how this holding, these are again cytoskeletons, extensions, and because of that, if there are gap junction, then what are the benefits of gap junction? If there are tight junctions, why there are tight junction? Okay, each has got their own functional utility. Then it attaches and it forms to tissues. Fungi, bacteria, and other cells, they have cell wall. It provides a mechanical support to the cell, and it precludes the passage of larger molecules. The selectivity, permeability is very important so that obnoxious chemical will not enter. It regulates it, and thus various types of transports are ever seen. Because of that, the molecules which are large, which don't have pore, cannot transport it. They are transported only through active. That means body decide which molecules to take inside the cell. If there are no transporters available, whatsoever good the things can be, it may not be taken in by the cell. Okay. Then second thing we have studied about the cell potential, which very important, particularly in nerve conduction and muscle contractions. So these are various mechanisms are there. Will not go for again various transport mechanism like osmosis, diffusion, etc. Then transmembrane protein channels are there, which are playing important role. Aquaporins we have seen recently in diuretics, that is only water are there. So all transmembrane transporters, I told you, who are interested in still more details, very nice chapters are there. Particularly in Goodman, very good chapter is there. All types of transporters, SLC transporters, ABC transporters, their classification, and how they are present, and I'll suggest you to read it once. Then next is we have seen endocytosis, exocytosis, etc. So these are all variety of functions these are making. So we have seen all these structures. We have seen fluid mosaic model, the polarity and their impact. Uh, then various structures we have seen about it. So this is how the structures are important. This is how the cell membrane is important. Now, mind when I repeat that all the components are made up, they are formed on from the genes which are there. You can just conceptualize this that from a two haploid cell when one diploid cell is made, that is from spermatozoa and ovum. When they come together, the first diploid cell is a zygote. And from zygote, we have got a body which is having around 300 trillion cells. Now, this much cells are present. Now, these cells again are going for except neurons and such. Most of the cells are going for the destruction and reconstruction or new formation, new generation. So this is what growth and repair you might have studied last year in pathology, tissue growth and tissue repair. Now that means this process is happening, and that preciseness, that selectivity is present with the genetic structure. That gene will guide about all these things. That what kind of uh, carbohydrate, what kind of protein, what kind of lipids should be contributed or should be present. Anything goes wrong, that will affect, that will make the impact on the function or structure of the cell. And you know, simple, very simple thing is that you know that if there is a alteration of structural or functional aspect of any cell or tissue or organ, then it becomes pathology. So in each pathology, the major contributor or the actually important contributor becomes the cell and its components. So this is what the importance of cell and components. Go through the cells and component. You also keep on reading about it, and it will help. Let us go for the other one, that is cell wall will not study, will directly go for endoplasmic reticulum, because we'll try to restrict most of the things related only to uh, human one. Okay, plant wise I don't think anybody will ask, and we should just remember because we are more concerned about uh, the human one. Okay, so let us go for that. You know about endoplasmic reticulum. We have seen two types, smooth and rough. Rough are because of ribosomes. Ribosomes form proteins. They are put into endoplasmic reticulum, and because of that, they are doing the job. The endoplasmic reticulum is not found in RBCs and spermatozoas. Otherwise, it is present in most of the cells. These are having many of the same proteins are there in the endoplasmic reticulum. Only difference is the presence of ribosomes and not. So they are engaged in the common activities, synthesis of certain lipids and cholesterol. Different type of cell, they contain different ratios of these two types of, that is smooth and rough endoplasmic, depending upon the activity of cell. The outer face of the, that is we have seen cis and trans portions of that. So we shall see, directly we'll go for the, these are the structures. I'll not go in more detail of the structure because you have studied it in physiology earlier. But you can go through, you can understand about it. These are the cis and trans portions, and these portions are there. They are comparatively very close to the nucleus, or almost attached to the nuclear wall, and the pores are there. Through pores, the genetic material, that is RNAs will come, and this RNA will get the activities done through the ribosomes, that we shall see for protein synthesis later. and then this endoplasmic reticulums, they will do the further job of attaching lipid or steroids and other things to the proteins. So uh, they are having various systems, cis and trans. We have seen yesterday also we discussed about it, and then they will do the functions are the synthesis and export of proteins and membrane lipids. Proteins, as I told you, proteins are actually synthesized by ribosomes, and they are just packaged or what we can say modified in this endoplasmic reticulums. Then next one, that is the quantity of both rough and smooth endoplasmic can in a cell can slowly interchange from one type to other. Means if the ribosomes are coming out and they are reduced, then it becomes smooth like that. Or if ribosomes are added, then it will become rough. Depending the change in the metabolic activities of the cell. It can include embedding new proteins in the membrane as well as structural changes. Changes in the protein content may occur with noticeable structural changes. It can this can means whatsoever protein which is synthesized from ribosome can be modified, and this modification can have a very big impact on the protein. So these are two reticulums, endoplasmic. One important thing is if it is present in a skeletal muscle, we call it as a sarcoplasmic reticulum. Okay, so instead of the cells are everything is there. Instead of simple thing, we call it as a sarcoplasmic membrane instead of cell membrane, and things are sarcoplasmic because muscle cell we give a separate name. Uh, Most of the things may be common, but there are something which are very typical because for the muscle contraction. Then next thing is what are these structures? We have seen that they are embedded. Rough means they are embedded with the ribosomes. The binding site of ribosome is called as a translocon. The ribosomes are not stable part. We have seen that they can be changing, they can be moved, and therefore it can. If they are moved, then it becomes smooth, and if it is there, then it will remain rough. The special complexes are formed within pre-ribosome begins translating the messenger RNA to the protein which is destined for the secretory pathway. First 5 to 10 amino acids polymerize, they causes encoding a single peptide. The molecular message that is recognized and bound to single recognized particle that is called as a SRP. Then translation pauses and the ribosome complex binds to the rough endoplasmic reticulum translocon site, where translation continues with the nascent, that is new, which is very newly formed, that is 5 to 30 amino acid we have seen now. That is there, and this protein is processed into the endoplasmic reticulum by an enzyme, which that is a single peptidase, which removes a single signal peptide, and this signal peptidase will remove it, and ribosomes may be released back into the cytosol. So they will be ribosomes will be now freely wandering in that. No translating ribosome are known to stay associated with the transloc. Okay, so this again will not go in more detail for the structures how it is. We have seen the cis and trans. It is a structure resembling a multi-story car park, etc. Will not go into that. What are the functions? We'll go for directly. That is, it is having the this manufacture of lysosomal enzymes with mannose 6-phosphate marker added into the cis Golgi network. Then manufacture of secreted proteins. They are either secreted constitutively, means always, or they are secreted in a regulatory manner involving a clathrin and paired basic amino acid in a signal peptides. So, again, if you remember nitric oxide, we have two types: constitutive and inducible. If you have studied earlier. So, constitutive means always, and inducible means whenever the cells are stimulated to do this, then okay. Then next is integral membrane proteins are formed because of through these endoplasmic reticulums that stay embedded in the membrane, and they bind to new membrane. Now, very important proteins are there which we shall be studying in the future course. These are the RAP proteins, SNAP proteins, SNARE proteins, and they are important in fusion events. They play a role in various uh, activities, particularly fusion, and therefore the subsequent activities from that. Then initial glycosylation assembly is there. The N means nitrogen linked glycosylation or oxygen linked. If the protein is properly folded, oligosaccharides transperases are the enzymes. They recognize the amino acid sequences, and accordingly it will add protein sugar backbone. Uh, this N-acetyl glucosamine, it is present in many. If you whenever you study penicillin mechanism action, you will find there are two important constituents of the cell membrane in bacterial cell, that is NAG, that is N-acetyl glucosamine, and NAM. So this that N-acetyl muramic acid. So like that, will have many such agents which are there in glycosylations. Smooth is already we have seen where no proteins are there because ribosome is not there. And third one I was telling about the sarcoplasmic reticulum, which is there in a muscle cells. So they are causing release of calcium induced because if you remember muscle contraction, then this muscle contraction is done after voltage gated channel. They are causing entry of sodium. Because of entry of sodium, calcium comes in. Because of calcium, from endoplasmic reticulum, the calcium will come out, and then it will cause more calcium will be there, and this calcium will cause contraction. This was what physiology of muscle you might have seen. So this is how functions are there. So this smooth is involved in synthesis of lipid and phospholipid and steroid. We have seen that the protein is not there. While in rough, proteins are there and they are embedded or they are mixed with the lipids or sugars that we have seen. So cells which secrete the products, example are testes, ovaries, sebaceous glands, abundance of smooth endoplasmic reticulum because only lipid-like secretions are there. Sebum is a liquid, and all the hormones, the steroid hormones, they are steroid in nature. They don't have any protein. So smooth endoplasmic reticulum will be doing this job. Okay, we have seen this all activity. What are various activities? So they are important in such activities. Then protein transport, that is embedding the protein into various actions. Okay, I think this should be enough for us. So these are the activities of endoplasmic reticulum. Okay, we shall see for next activities during next class, and you can put your difficulties on the WhatsApp group. Okay. Now here no presentations are there. I'll not be able to give presentation. Whatsoever presentation are there, I'll put into the this Google Classroom.